Thank you so much, Tom, for the invitation and for this introduction, which was wonderful. And good afternoon to everybody. Before I start, I wanted to say that even if at this time it's me presenting this work, I'm about to present the results of various research collaborations that I have with many researchers, not only here in Portugal, but also in Spain, in France, and in other places. And that's why I would like to acknowledge all these contributions to all those people involved. Even if their name is not explicitly here, they are part of this research, and without them, it couldn't be possible. Also, please, before I start, know that some of the images I'm going to show are a bit sensitive in content, not really creepy, but so please keep that in mind, and I apologize in advance if someone get a bit more sensitive to them. So for this presentation, I'm going to focus on birds, but first of all, I would like to do a small introduction about the role of small game, and which encompasses a wide variety of species, not only including birds, but also mammals, birds, reptiles, and other aquatic resources such as fish, shellfish, etc. Their presence in the archaeological record provides valuable insights into past human activities, and that's why even if they are not focus of attention for so long, now they are start having their importance in, especially in the Paleolithic archaeology. To contextualize a little bit the story of investigations about small prey in general, it is important to talk about the theoretical ecological postulates established to interpret how predators choose their prey. One of these postulates is the optimal foraging theory, which helps to predict how animals behave when searching for food and considers only the energy provided by the prey as well as the handling time to get them. This model assumes that based on the optimization of resources, animals will maximize the amount of energy gained per unit of time, energy, and risk involved in finding food. Thus, it assumes that foragers in general should ignore low-rank animals if more profitable prey are abundant and available in the environment. For some time, this model has been applied to human subsistence too. And in this line, only in moments of scarcity of more resolutive resources, humans would opt to exploit less profitable animals. This was proposed to the increasing of a small game that was perceived in faunal assemblages at the end of the Pleistocene and the beginning of the Holocene, which could suggest some way that there were some changes in hunter-gatherers' behavior that pushed them towards broadening their diet. Thus, there was this general assumption that small animals passed unnoticed for human populations for so long. And also for this reason, most of the faunal studies made on these taxonomic groups were limited to their identification used for paleontological or paleoecological inferences. But no studies at all were developed to study bone surface modifications produced on these animals, as it was assumed that they were not used by human groups. However, the incorporation of a small game in human habits has to be perceived not only from a nutritional perspective, as many other products can be obtained from this prey. For this reason, for example, the theoretical framework does not always fit with archaeological reality. We should take into account, in fact, that human behavior may be unrelated to energy maximization. Also that social cultural changes are less predictable. And of course, secondary products not always related to nutritional purposes may exist. For all these reasons, during the last 2 decades, new research has been developing, developed, trying to revise some small game assemblages more in depth. Proof of that is that they were commonly used by prehistoric populations, as you can see in the map. For example, in the case of birds, we can see that many European sites present evidence of bird processing in Neanderthal context. In fact, this map that you can see here was published in 2022, but it's already ancient because we have already some new data that is not in the map that prove that there is an increasing number of sites recording the use of birds as well as other small game through prehistory. But the use of small prey, and particularly birds, is not only limited to what we call classic Neanderthals, as we have some evidence that is really old, and it goes back more than 120,000 years old. However, when we are talking about birds, we are facing the study of a wide variety of taxa, each of them with some specificities that may affect the way they are procured, processed, used, or even abandoned. And as archaeologists, we do not always understand how these animals may have been used. And that's why some of the marks we observe on bones, or even the absence of them, are difficult to interpret. In this context, experimental archaeology provides tools that help to interpret the archaeological record. First of all, it can provide, based on direct experience, different potential ways of processing birds. It also offers insights into intuitive gestures used during animal processing that might otherwise go unnoticed. And in addition, it helps to associate the specific gestures or activities with bone surface modifications or some representation patterns. Concerning birds, up to date, different experimental work has intended to associate activities with modifications observed on bones. Most of these works were carried out on some small or large sized birds using common taxa found in archaeological sites. And some other works also focus on the processing on large birds of prey. But even within the category of what we call medium-sized birds, significant differences can be observed. The anatomical characteristics of each bird, such as the robustness, the weight, the fat content, or the muscle mass, may vary considerably. And depending on the intended use, certain birds may be preferred over others. For example, if the primary goal is to obtain meat, a more fleshy bird like a rock dove would be a better choice than a rook, right? And what it's true is that one of the problems of facing a bird archaeological assemblage when we start analyzing the bones is that avian bones often shows minimal traces of human activity because they are small, fragile, and can be consumed using only hands and teeth. Unlike large mammals, birds can be easily broken, chewed, swallowed, leaving very little evidence of human tools, of human traces. And in fact, some experimental studies have been investigating some of the bone surface modifications beyond the traditional cut marks that could be linked to human activity during bird carcasses processing. These include holes, fractures that may result from the overextension of the elbow joint, as well as other breakage patterns associated with the separation of the wing bones. However, research in this area is still scarce and on its early stages. Also, some studies have attempted to identify human chewing on bones, not always with clear success. Also because distinguish between these traces and other traces produced by carnivores, for example, can be really challenging. Additionally, while some research has focused on recognizing human chewing on bones, most of this research has been conducted on larger fauna, with little attention given to small game and no studies on bird primates. This absence or negligible presence of traces on bird bones raise the real need to tackle the problematic of how to distinguish human processing patterns on birds. For this reason, we have performed experiments together with some Portuguese colleagues, Mariana Navais and Marina Igreja, to document what kind of actions occur during the processing of small-sized birds and which of these activities generate some kind of bone surface modifications that could be perceived and diagnostic. The first experiment we performed with our colleagues was carried out in two different phases. The first phase is already published, but the second one is still undergoing, so I'm not going to present the final results here because it's not ready yet. But in both cases, we try to focus on the mechanical removal of meat from species commonly consumed by humans during prehistoric times. Here you can see the first phase of the experiment where we work with 5 wild birds, and the second part of the experiment where we were working more on not domestic animals, but animals more commonly in archaeological contexts. So for both phases of experiments, we conduct experiments at Ecored Lab in a controlled research environment. For the first experiment, we selected 5 avian specimens, including 2 colored dogs, 2 carrion crows, and 1 wood pigeon. These specimens were recovered from the Wildlife Ecology Rehabilitation and Surveillance Center, which we call SERVAS, located in northern Portugal. And for the second experiment, we selected 2 partridges and 2 anatids, commonly found also in Iberian archaeological contexts, and they were also legally acquired in contact with farmers and local producers. The initial conditions of birds were documented before the experiment, and manual techniques were used during the battery process. A flint tool was used to help with some of the actions, and then bones were cleaned and analyzed under the stereo microscope to document any kind of bone surface modification. The processing of both raw and roasted birds follows similar actions. All birds were firstly manually defeathered to facilitate the process. Then 3 of the birds in the case of the first experiment were roasted, and the second one was 2 of them. All individuals were manually disarticulated and defleshed, raw or already roasted. And the gestures were, first of all, removing the head by twisting and pulling from the cervical vertebra, then trying to remove chest, skin, and meat manually or by using a flint tool when necessary, detaching wings by twisting and overextending the elbow articulations and then the fleshing, and then legs were separated by manual twisting, flexion, or overextension of the knee articulation. For those animals in raw state, it was really difficult to separate the meat from the bones, so the use of a flint blade was necessary. For the second phase of the experiment, as I said, we are still ongoing on the analysis of the remains, but we can already give some information. For example, the same protocol was followed as in the first phase, but however, on one of the ducks, it was not plucked, but directly skinned instead, allowing for the removal of the entire feather corpus from the body. It took more or less the same time because it was quite a big animal, but it was a bit more easier to remove the whole corpus of feathers instead of going one by one. Partridges, for example, were processed very quickly manually, even when raw. And in the case of ducks, especially regarding the wings, a stone tool was also required to remove the feathers. A tool was necessary for disarticulating raw birds, of course, and processing certain parts of the duck chest when it was already cooked. So for the first phase of the experiment, long bones from 5 individuals were analyzed to detect any potential damage. On uncooked specimens, cut marks resulting from disarticulation and the fleshing process were observed near the articular joints, where tendons and ligaments are more attached, and also on the shaft of the bones. Wrenching marks, as well as notches or squashing marks, were also documented in relation to this overextension of the elbow and wrist joints that caused the breakage of the bones. In the case of roasted birds, the only bone surface modification observed in almost all or almost all the bones was burning damage. The distal elements of the extremities, including wings and legs, were completely charred and often lost or severely damaged. And if we look a bit more close to the skeletal representation of these animals, we can see that on uncooked specimens, they preserve 100% of their skeletal elements. On the opposite, roasted experiments exhibited a predisposition to bone fracture and loss, and almost 60% of the bones disappeared due to burning processes, especially the distal extremities, both in wings and legs. So to conclude with this first experiment, we have been able to confirm that bird processing can mainly be made manually or with the help of a cutting tool in the case of uncooked specimens. However, in the case of larger birds such as ducks, a tool is also needed even if the animal was previously cooked, potentially leaving also some traces on bones, especially the sternum, which is the part where a tool is necessary to remove the meat. On the other hand, bird bones break while being processed due to twisting, flexion, and overextension of the articulation joints. But, however, any other kind of tool, apart from the stone tool for cutting, was necessarily used. I mean, in terms of we never use any hammerstone or anything to break the bones. Particularly in the case of uncooked birds, we can find cut marks, perforations, and notches on bones because of the resistance of the tissue to manual separation and the need for more abrupt movements and the use of tools. Okay. But on the contrary, also due to the no need to use hammers, other damage related to percussion, it's not present, like percussion marks, impact flakes, etc. Also, in the case of bones from roasted specimens, they show burning stain due to the exposition of the bones to high temperatures. And in relation to that, bone loss occurs due to the roasting and processing of the roasted animals because they are more fragile and the cooking process exaggerates the bone loss at some point. Similar patterns associated to food procurement observed in these smaller birds are extended to other large animals, large birds, such as in the case of birds of prey. In fact, archaeologically, we have some evidence of birds of prey that were potentially used for food. Okay, here you can have in this slide, you can see some examples of them. However, we have to say that the evidence show that there are presence of cut marks on raptor bones that cannot be linked to meat extraction itself, as some of these cuts on wings and talon phalanges are located in areas with little to no meat. So in that parts, only tendons, feathers, or scales are present. And this would suggest that hunter-gatherers had an interest in these parts for other reasons than food, right? And in fact, we should keep in mind that birds of prey have long captivated human societies due to their powerful and majestic nature. Ethnographic studies reveal that many cultures, even today, still admire and respect birds of prey, incorporating them as part of their rituals or traditions. And this suggests that the potential cult of eagles or other raptors could have been done even in the past. Moreover, there is also a tangible living connection between humans and birds of prey. Many raptors found with evidence of human activity archaeologically are either occasional or regular scavengers, meaning that they frequently interact with human settlements at some point. And why not this proximity might have potentially encouraging some kind of connections among them, right? And it is also very important to consider when analyzing these bird of prey bones that even in areas of raptor carcasses where cut marks are found, but no meat is present, these regions are also correspond to places where we have primary and secondary feathers strongly attached to the bones, which are difficult to remove manually. As a result, the presence of cut marks in these areas may indicate intentional feather extraction. Long tendons are also present in both wing and leg elements, which cannot separate without the help of a tool, right? In this line, we try to perform with some colleagues from Pasea Lab in France, Dr. Véronique Laroulandy, and one of our students, an experiment to create and interpret reference collection that could help to understand the traces observed on archaeological materials. notably on wing bones, but also on foot phalanges. Okay, particularly on the penultimate and the talon. The goal, in fact, was to decline the type and number of cut marks produced and also the associated gestures within this process. As a result, a total of 10 raptors, both diurnal and nocturnal birds of prey of various sizes, legally acquired, were used for the experiment. Small raptors were excluded from the talon extraction since they have no meat. They have not been found in archaeological record. So as they are not commonly found, we just exclude them from the processing. Also because it was very difficult to separate, to manipulate these elements. The animals were processed, all of them using unmodified clean tools. And once the experiment was completed, as in the previous one, the skeletons were carefully prepared to remove soft tissues while ensuring bones remain intact. And observations of the modifications on the bones were conducted the same using a stereo microscope with magnification up to 45. For feather removal, we perform longitudinal cuts along the wing to separate the skin and the feathers. Additionally, circular movements around the proximal humerus were performed, and small incisions into the carpal, metacarpal, and the phalange regions of the wing, the end of the wings, were also common to detach the feathers, mostly firm attached to the bone. Shorter cut has been also made to separate the muscles and the tendons in the regions of the elbow joint. For the talon extraction, the talons were separated from the rest of the foot at the joint with the penultimate phalange. Mainly transverse and circular movements were made around the joint. And in some cases, an incision was made on the plantar side of the first phalange to release the tendon and facilitate the process. As a result of this analysis, we consider different areas to localize the marks. And we consider also the presence of striations, their quantity, as well as their location and orientation. And if we make a general assessment of all the wing bones, we find that almost 66% of them show at least one striation. Nocturnal raptors have a slightly lower percentage of striations, but this difference is not statistically significant. The long bones and the first phalange of the major digit of the wing are the most affected bones, as you can see here. And in 2/3 of the cases, the striations appeared root. Additionally, oblique striations are the most frequent, followed by longitudinal striations. In these cumulative diagrams that I'm showing right now, you can observe all the striations that have been identified on the humerus. On the proximal part, the striations seem to result from circular action performed to separate the scapular feathers from the rest of the wing. There are also striations on the body of the humerus, which are the consequence of removing the skin and the cover feathers. And on the distal parts, there are also striations that may be related to cutting the muscle insertions and movements at the elbow joint. This last marking echoes those observed on the proximal part of the ulna and also on the radius. These bones also display striations on the body, especially on the anterior and posterior surfaces, which are related to the oblique movements performed to detach the feathers attached to the bone and to cut the skin. For the lower part of the wing, for the carpal, metacarpus, and the phalanges, striations are found on the proximal part of the carpal metacarpal at the joint with the first finger, and on the main shaft, where primarily feathers and the skin are really strongly attached. On the phalanges, we can also observe some striations continuing these similar actions performed on the carpal metacarpus. Regarding the legs, as you can see, more or less 79% of the distal ends of the penultimate phalanges and 82% of the talons show at least one striation. Approximately 60% of the striations are root and mainly oblique or transverse. And no notable differences were observed between the processing of diurnal or nocturnal birds of prey. However, if we look at the number of striations observed, we can see that certain taxa exhibit remarkable values. This is not because of the taxon itself, but we thought it's because these animals exhibited high rigidity and contraction while processing them, which require a certain level of persistence to detach the talons from the specimens. Most of the striation groups are located of course, on the distal part of the penultimate phalange, but specifically on the lateral, medial, and articular sides. For the talons, all the striations are located also on the proximal part of the talon, and mainly on the lateral and medial faces, and also on the flexor tubercle. And well, if we start comparing the overall results of this experiment with those obtained in other experiments that other colleagues performed, we can observe that they are relatively similar. All of them present a high percentage of bones with cut marks. Also, striations are mostly oblique and longitudinal on the wings, and oblique and transversal on the phalanges, on the distal phalanges. Bones like humerus and ulnus have multiple striations, while radius show fewer marks. And in the case of Carpometacarpus, they are also heavily affected. In the case of our work, unlike other works already published, these one do not show breakage or peeling patterns on the Carpometacarpus, as we did not force the joints at this point to separate the feathers. This also explains why we have presence of cut marks on the wing phalanges that are not found in other studies, because they just break this portion to get the feathers. Finally, the penultimate phalange and the talons are heavily altered on the lateral and medial surfaces. And what we can say is this work added value of our reference in the sense that the number of observations we made is higher than in other previous works done. Most of the works done until that time were using a maximum of 4 specimens, 4 individuals were processed. And in that case, we used for the wings 10 individuals and 7 individuals for the phalanges. As a result, we also recorded a higher variability in the traces produced. However, the most important aspect of this research is that the traces observed are comparable with in many cases with those found in the archaeological report. For example, in the case of the MIS 6 layer of the Lazaret Cave, where a golden eagle humerus was found with marks on the distal area, as you can see here. And if we look at the results we found in our experiment, also the humerus is a bone that still have a lot of meat attached. These marks are similar to some of those observed in our study, and might relate to the separation of the tendons and all the elbow joint. The same applies to the cut marks observed in posterior layers of Humane, where we found some wing elements and talon with comparable marks. And as you can see also, we have these traces very close to each other. The marks observed on the penultimate phalanges and talons of raptors in Crapina are also very similar to those documented in our experiment. In fact, it can be deduced that the movements performed to separate these elements would have been similar, or not the same maybe, but really close to each other. Here you have a comparative example. The same applies to Mandran, where also raptor talon was found, and we can find the same traces on the tuberculum or on the both lateral and medial side of the phalanges. And the same applies in the case of the talons found at the Mousterian sites of Combe-Grenal and Le Vieux, where these traces, especially on the dorsal part of the talon, are also documented. So, as a general conclusion, what we can state is that the presence or absence of bone surface modifications on birds depend on a wide variety of factors that can include, for example, the reason for acquiring the animal, whether for food, to make tools, or any other purposes. In addition, the animal may display a wide variety of cut marks, some of which overlap with different activities performed during processing, such as butchering, skinning, etc. Also, the manner in which the carcass is handled and processed will also influence on the type of bone surface modification we will see. And the presence or absence of meat in the activity areas where we are focusing can also impact the type of modifications we will find. Whether the meat was cooked or not will also affect the way the bones are modified, as cooking can make bones more fragile, and after the marks can be very altered or invisible. But at the same time, cooking would facilitate the process of manipulation of the animal without the need of using tools. So there are some pros and cons, right? Also, the skill level of and the knowledge of the individual processing the animal is very, very important, because it's not the same that someone that has some knowledge about how to process the animal or the anatomy of the animal may have potentially less traces than another that doesn't have any kind of knowledge in that sense. Also, other factors, of course, such as the type of tool used during processing, the age of the animal should be taken into account, which could result in variations in the bone modifications observed. For all these reasons, I think it's crucial to develop a corpus of experimental series in order to figure out this variability in the kind of traces we can observe on bones. And it's also important to develop this extensive experimental, reusable, and accessible data in a standardized manner, so we can access data from other people in a more open way. In this line, in fact, I have recently read a paper from some zooarchaeologists that put a lot of emphasis to this approach, and I found it particularly insightful, because as in zooarchaeology, ethical considerations are of utmost importance, as the treatment of the animal, the methods used in experimentation, the interpretation of the results must always be conducted with care and respect. This will ensure that our research is not only scientifically rigorous, but also socially responsible. And just to conclude, that it's not related with what I have explained before, but I would like to do a specific mention to a new exciting experimental project that we are starting with Professor Cristina Real from the University of Valencia, and also in collaboration with other colleagues from ICAREP and Spain, where we want to focus on the different cooking and preservation techniques potentially used for rabbits. Our goal is to see this, if this has a parallel on the archaeological record. And well, for now, we are really excited, and we hope, we are very optimistic, and we hope to have very promising results very soon. Let's see, maybe in another seminar. With that said, thank you so much for your attention, and I am open to questions if you have any.